Last but certainly not least, our third and final speaker is Dr. Michael Dennis. Michael is the president and CEO of Performance Genomics Incorporated, and as many of you know, was also a former president and CEO here at Genome Atlantic. So once again, someone we've known for many years. Thanks for joining us, Michael, and the floor is all yours, sir.
And thanks for the invitation. Pleasure to be here. Tell you about Performance Genomics Inc., PGI. PGI is part of a revolution that's happening in agriculture, specifically in livestock breeding. As you know, livestock breeding has been used for millennia to improve progressively the genetic performance of livestock herds. Through the quantitative analysis of adult offspring, to looking at how adult offspring perform, to be able to pick best breeders to improve the herds. The revolution that's happening now is allowing livestock breeders and producers, rather than waiting for adults to determine how well a breeder does, analyze the genome sequence of newborn livestock and using algorithms developed that can convert the genome sequence into predictions of future performance, select those animals and only those animals with high potential to raise within the herds and to produce the next generation of livestock. It's moving very quickly. It's heralding really a new age of genomics in applications, and I'm calling them genome apps. We're going to have a set of genome apps that can go in, look at a sequence, identify specific markers that have importance for specific traits, and make quantitative predictions that can be used in everyday decision making and management in livestock production. Where do we fit? Well, PGI has a unique asset, biological asset, developed over 25 years, a mouse colony that produces twice as many litters and has twice the reproductive lifespan of a control mouse. Genes in that mouse have now been selected over time to give those mice superior performance. Our objective, identify those genes in the mouse, use them to translationally into other mammalian species, specifically livestock, to identify those sequence differences that are responsible for performance differences, specifically within the area of reproduction. So our objective, a genome map to predict a reproductive lifespan of livestock and to allow dairy producers or livestock producers to progressively improve by selection the reproductive lifespan. This revolution is being driven. We've heard a couple of references to this revolution in sequencing that is driving really the practical applications now of genomics into industrial life and in agriculture. The sequencing explosion, Steve referred to the $3 billion and 10 years that it took to do the first human genome. In the period, the 12 years since, a genome has come down to under $5,000. We see $1,000 here from $3 billion, and we're talking about 2 weeks instead of 10 years. Million-fold reduction in cost, hundreds of times faster, more powerful, much cheaper. So the application is being driven by reducing costs. It's possible for a dairy farmer, for example, to test his cow for $40 now for a specific set of markers that allow him to select the cow for better traits. The other thing, obviously, is that reduced sequence is allowing scientists, the industry and the academia, to sequence essentially every commercially relevant species, the genome of every species out there. Humans started 2012, chickens followed, mouse and cattle in 2009, 2 weeks ago swine. And sheep will be next year, and goats will be the year after, and we can expect in 5 to 10 years, all of the commercially relevant species will have full genome sequenced. They're sequencing genome of Nova Scotia mink at the agricultural faculty in Truro right now because it's an important species for the region. So that's driving really the appearance of this genomics technology within everyday life. And if I could put it in context for our target, dairy farmers, have been working for hundreds of years to breed that perfect cow, which would have a good humor, stand comfortably, have long health, good fertility, obviously good milk production, but at the end of the day would be profitable. And increasing profits is still the primary driver for dairy producers. How do we increase our profits? Using traditional breeding, selective breeding, turbocharged with artificial insemination, which is 90% of dairy reproduction now is artificial insemination. It was possible to increase over a period of 30 years by 2 to 3-fold milk production per cycle per cow. So incredible success in increasing that aspect of dairy cow performance. But in parallel, other things declined because they weren't understood. Fertility in particular progressively declined. Where cows used to last 4 to 5 cycles, they now last 2 to 3. A dairy farmer with 100 cows needs to have 80 cows non-productive just to supply 40 per year to replace the ones that are culled every year. So there's a 40% cull rate. Increasing by 1 cycle, 1 more pregnancy would cut $15 billion cost out of the industry in replacements, the global industry, and add 10 to 20% to the bottom line profit for a dairy farmer in Canada. That's where we are. We have a set of, we don't have our genes in mouse identified. We have our platform in development. We're applying the latest tools to the identification of those genes. We've identified and signed an agreement with a major partner that is already out selling genomics tests for dairy cow selection. Over the next 18 to 24 months, we'll be mining our mouse and transposing that information into dairy cattle genome with the objective that within 2 years, our partner will put those tests, new tests, in the hand of farmers to allow them to select breeders that they'll end up with herds that have fewer numbers, but they'll be more profitable with fewer, healthier, longer-lasting cows, which is really a practical and very concrete application of genomics, which is already in the marketplace. So thank you.